Oh, never mind. Hi, chat.
There's chat. We believe the chat.
Are we missing anybody else?
I don't think so.
Okay, just
Okay, so restricted enzymes, y'all remember all of that?
Where's my book bag?
There's tons of them, hundreds have been found. Most of them that we use in biotechnology produce sticky ends, but not all of them cut like that. Some of them cut straight down the middle, and those produce what are called blunt ends. They're not really of much use in biotechnology because we use the sticky ends to allow them to base pair with maybe another piece of DNA from another organism. So anyways, there's lots of them. Some of the most common are EcoR1, HEN3, BAM-H1, SMA1. Sorry, there's lots. Anyway, these are the guys that found them first. The first one, EcoR1, restriction site GAA-TTC. You don't have to know that. So they cut specific, and these recognize the palindromes, most of them, not all of them. And then we'll cut in between two of the same nucleotides. When they do that, they produce pieces, and the pieces are called restriction fragments. Like I said, they're not much use like that necessarily. However, we can use restriction fragments for analysis, for forensics, and for evolutionary relationships and all that. If that's the case, you can use the ones that do blunt ends. So if you're trying to mix between different organisms or add genes to something, you need the sticky ends. So if you have another piece of DNA, you want to add a gene into like a plasmid, for example, you have to cut both the plasmid and the gene of interest with the same sticky end. So they have, or the same restriction enzyme, so they have the same sticky ends. So that then when you mix them, they could hydrogen bond together, right? If they were cut with different restriction enzymes, they would not be able to do that. And then you could just use ligase to make the phosphodiester bonds. And now you have a piece of DNA that is from both this yellow organism and then the one that is blue. We call this mixture of DNA recombinant DNA. It's a new mixture that has never existed before. In this case, we would, a lot of times, like what we're going to do in our lab, use a plasmid that has the GFP gene on it. And now you can use a plasmid as a vector to take that gene into the bacterium and allow it to transcribe and translate it to make the protein.
So, like, if you did the things that they, like, those lids, I would pass on.
Yeah. The way they did it, yeah. When later on, we're going to talk about using something called CRISPR, which is gene editing. And it's sort of in its new stages. It's very new, but using CRISPR, a lot of times, using CRISPR gene editing, now it's very different from this, but it is not ethical and not done on germline cells, which means cells that, so they can't be passed on. So right now, if they do CRISPR stuff, it's on somatic cells, so it won't be. There's a lot of ethical issues, making designer babies and all this stuff that is not legal. Yeah, they're using CRISPR stuff. The thought is to do that on someone that has sickle cells, to take out the damaged allele and put in a normal allele, or something like that. So yeah.
But wouldn't they have to do that on every single person?
Yeah, so the sickle cell would be just in the hematopoietic stem cells, so in the bone marrow. Yeah. So not every body cell, but the ones that will make that particular protein. We'll get to that later. Okay.
Didn't we talk about this already? Like we did our bacteria chapter. Like I feel like we went to this.
We talked about transformation. Yeah. I'm combining them now.
Okay.
So why do we care to mix genes together from different organisms like a bacteria? So what's the use of bacteria? Yes, I've already talked about this a little bit. But this is the chapter where we do this transformation lab. It's where you'll actually be tested on the usefulness of stuff about it and the process of it. So in this case, this is a picture I think in your book. So here's our gene of interest inserted into a plasmid, and then allow the bacteria to take it back up through transformation, allow the bacteria to make many copies of itself and therefore many copies of the plasmid. This process of making many copies of itself and the plasmid is called cloning. You're producing clones of the plasmid. Now, why do we care? We can make many copies of the gene itself, And then isolate the gene. Oh, Jack, you okay? I'm so sorry. Have you been here? Or have you been at the doctor?
Oh yeah, I went to the doctor.
What did you say?
He just said it's pregnant to stay on crutches for a week.
Well, we're recording for you. Oh no.
Oh, you need a replacement?
We don't really use this process anymore, however, to make copies of DNA. We use another process called PCR, which is much faster and much cheaper. We'll get to that later. The better use of this now is for producing the protein, so allowing the bacteria to transcribe and translate that gene to make the protein. You can do basic research on it, but more usefulness is to purify that protein, and this is like human growth hormone, use it for insulin. That's how we actually make a lot of that stuff. And we'll talk about other stuff later on. So now, let's go back for a second. If this picture was actually accurate, and you took this nucleus from a eukaryote, isolated a gene, and stuck it in the bacterium, would it actually work? Would it produce a functional insulin protein?
Yeah.
If you took the DNA right from the nucleus?
Yeah.
No. Well, there's systems. What? You're thinking of something else. Why would it not work? It wouldn't produce insulin. Not like this. It has introns and exons, and bacteria don't do splicing. So this is actually not the way it works. You have to do something else. How can you deal with it? How could you, hold on, we already talked about this stuff. We already talked about this. So how can we clean up the introns? Slice it yourself? Let the cell do it. Let the cell slice it, but then what do you have? Messenger RNA. Yes, you start with the messenger RNA. So what you do, no, you can't insert mRNA because it's RNA. You can't do that. So then you use reverse transcriptase to turn it back into DNA. Yeah. So for eukaryotes to do this process, you actually isolate messenger RNA from the cytosol. It's important to take it from the cytosol because if you take mRNA from the nucleus, it might not have been spliced already. So you have to isolate mRNA from the cytosol because you know it's already been spliced. And then you have to go back to DNA using reverse transcriptase. Reverse transcriptase will take it back to a single-stranded DNA molecule. This is all. I'll send you this. I sent you this already. And then you can use DNA polymerase to just make a copy, and then you have your DNA, your gene without the introns. We call this DNA cDNA or copy DNA. That's the type of DNA then you can isolate or put into your plasmid, and then the bacteria can go ahead and transcribe and translate it and make a functional protein. I'm going to pause for a second and tell you this. I have 6 PowerPoints. I will send them all to you. If you study from my PowerPoints and study from the book, your test is next Friday. My PowerPoints are kind of out of order from the book. And I'm a very sequential person. I like to go in order when I do things, but to me, the way I organize these PowerPoints makes more sense. I insert applications, like why we use this stuff into these PowerPoints, but your book, it does sequencing first and it kind of does some of this stuff and then has all the applications at the end. And mine's organized differently. And your chapter actually starts with sequencing, which I don't talk about till like the 4th or 5th PowerPoint. I'm just telling you, mine are not in the same order as the book, but all the info is there. Now I'm actually including extra stuff that's not in your book, like when we do CRISPR and some other stuff that is not in your book, but it's important to just know about it. It's very new technology that you'll see all over the news. If you go to college, you'll hear about it. So might as well talk to you about it now. No.
Good.
So man, if you wrote about that on NPDM response, I was like, what? You gotta explain. Alright. I mean, if there is a question. Okay, so, and your test is next Friday, by the way. I think we're going to do this transformation lab. I'm not sure when it's coming. If the lab actually doesn't get here until Wednesday, then we can't probably start it until Monday, which would be good for y'all going up. We'll see when it gets here. Anyways, and then the Gene Machine people from the Greenwood Genetics Center are coming next Thursday. So it's just going to be in your class. You're not going to do anything extra. But during that time, and they're going to actually do it in here. We're not getting on the bus. The bus will only hold 20 people. And I have 2 classes now, and both is definitely bigger than 20. This is the first year they have a mobile van. They can bring all their equipment and come up to a lab. So I was like, yes, let's use it. So anyway, they're doing that next week, and you'll be able to do PCR. What?
Watch through them, don't hold them.
Yeah.
Well, they won't have the van. They won't bring it. So anyway, that's next Thursday. Moving on. Okay. So transformation, bacteria. So the way transformation works is you have your, let's go back to the picture. Whoa. So you have your plasmid that has your gene in it, and you have to allow the bacteria to take that up. Well, how that works is you have it in broth, right? And you stress it out. So you have to heat it up a little bit and expose it to a chemical, and the bacteria will open up channels to bring in external DNA. But not all bacterial cells will take up your DNA, your plasmid. Not all of them will. And so how do you know which bacteria have taken up your plasmid and which bacteria haven't? Because usually they'll look the same if you grow them on agar. How do you think you can select for them?
Something that definitely shows that it's been selected on that one.
Yeah. One of the really easy ways to do that is in your plasmid, you also have a gene for antibiotic resistance. And then grow it in the presence of an antibiotic. So ampicillin, for example, is an antibiotic. So if on your plasmid, you have a gene also that codes for ampicillin resistance. And then you grow the bacteria on agar that has that antibiotic. Well, the only colonies growing will be the ones that have your plasmid. The rest of them will die. Does that make sense? That's what you're going to do in the lab when we do it. And I think it's ampicillin also.
Is that the luminescent?
Mm-hmm. Our DFP lab. Last year, every group works except for one.
Was it their error or?
How's something they did wrong?
Yeah.
This lab is foolproof unless you do something wrong. There's a lot of steps, and if you don't do it exactly right, it's probably gonna mess up. Okay, most of the time you can buy plasmids already made. And then, so in most plasmids you can buy, there's something called origin of replication over here. That's so it can replicate inside of a bacterial cell. So when the cell divides, it can have lots of copies of the plasmid. Here's our antibiotic resistance gene. And up here in the other part of the cytoplasm, there are different restriction sites. Usually there's multiple restriction sites. Because if you're going to buy a plasmid and you want to insert a particular gene, you want to buy a plasmid that is most useful. So you can choose whichever restriction site will work the best for you. Anyway, so that's what these little black markers are, different restriction sites. So if you cut your gene from whatever organism you want to put in there with equal R1, then you would cut this plasmid with equal R1. And then when you mix them together, they have the same sticky end. And it would just be stuck up here. So, are y'all good what the selectable marker is and all that? We're good? Okay. Like I said, this is a very important part of the AP exam. If you're going to see anything, usually in biotechnology, it'll be this lab, it'll be gel electrophoresis or PCR. And we do a whole lab on this. And you will do gel electrophoresis next week with the gene machine people come, and they'll talk about PCR also. Okay. So we have a whole lab on this, and we're going to do, we probably have time. What that stuff is up here is they have a paper lab where you create your own plasmid, and that's what we'll do that later today. So let's talk about some benefits of using genetic engineering. So you can create genetically modified organisms. Scientists have made a lot of them. One particular type that is out there is called BT corn. It has a gene inserted in the corn genome from a prokaryote that produces a particular toxin that will kill this corn borer, this little caterpillar guy. The corn borer is really bad for farmers because it will eat their corn crop, and then they can't sell it, and then they can't make money, and then they can't feed their family. So This corn is useful in that it helps protect their crops from one of the types of pests that could eat it and prevent them from making money. Another type of genetically modified organism are fish berries. It'd be weird if you went to the store and you saw fish berries instead of strawberries. But they're strawberries that have a gene from a flounder that is lured of a an antifreeze gene. It helps extend the growing season of strawberries to much earlier. You can start earlier. It helps protect them from frost. So then they can produce strawberries for a longer period of time. So if a gene from a fish is inside, no, they don't at all. They taste like strawberries.
You're terrible.
It's the gene from the flounder, not the flounder.
Let's see. All right. So golden rice, we've already talked about. The vitamin A has been added. Let's see. In certain strains, I have some other stuff on here. In beans and fungi, and wild rice have been modified to have iron in it. Let's see. There's lots. There's some pineapples that have a gene from rice that are resistant to nematodes. And so,
Those are the ones that eat SpongeBob.
so what's also important about these pineapples, for example, or this corn, is it also eliminates the need for using certain pesticides. And so because they're naturally resistant, they have their own internal pesticide, you don't have to spray them. So one is cheaper for them, they don't have to spend the money on the chemicals, but also now you're not consuming as many chemicals when you eat the food. Does that make sense? Now there are some concerns, there's lots of GMOs, you're going to read an article about it later, but there's some concerns regarding the use of genetically modified organisms. Now all the research out there says they're very safe to consume. So most of the time if you see stuff talking about concerns, they're worried about food for consumption. Some of the things, someone might have an allergy that is unknown to this toxin that is made from the bacteria that protects it from the BT corn. Someone might have an allergy to that toxin, but you don't really know unless you consume it. So unknown allergies is one of the concerns. Another concern is creating organisms that are like super plants or super weeds that will outcompete now in a natural ecosystem. That might be nothing. And if you're modifying them and so make them for whatever reason able to outcompete, they can produce yeah, so they can now outcompete everything else, and when you do that, it reduces biodiversity. You know what that means? The number of different organisms, and why is that bad for an ecosystem? Yeah, so you remember the potato blight in Ireland or the Dust Bowl? Didn't y'all learn about that in some history class? They were doing monoculture, depleted the soil of certain nutrients, and then nothing else could grow. That's kind of the thing that happens with invasive species. So anyway, this stuff is in your book. We'll come back to it a little bit later. So we do a lot when we make our GFP bacteria. Hello. All right. So you can cut DNA with restriction enzymes. You can paste it with ligase. Copy, how do we make copies of our DNA? What was one place, we took one method using the plasmids of the bacteria just copying themselves and therefore copying the genes. There's a much easier way. It's called PCR, and we'll talk about that tomorrow. We haven't talked about how to find DNA at all. So that's the stuff we talked about so far. You know what I'm going to do, just so we are very consistent. We're going to go ahead and do our paper lab since we're already talking about that. We're just going to do it now. So, and then once we finish, we can move on. All right. This lab, when I copied it this morning, I accidentally included 2 pieces of paper on the back that you don't need at all. So here's a waste of paper. The last 2 pieces of paper you can just rip off and recycle.
Olympics. Oh, man. It don't cost nothing to pay attention. Oh! That should have been.
The last two pieces of paper that say print on green paper, that was an accident, just take them off. This lab is super short.
It said on my home 2 pieces. Really clean rep. All inside of one. It's not like mine where
Is it being recycled?
Yeah.
I'm gonna turn the frame.
You still want it.
Everybody needs one green and one white. If I don't have enough right now, I'll give you one in a second. I got one.
What are my green though? Yeah, but I had
Are you serious? Swear to God.
Yeah,
I need it for a whole day.
Yeah, I got too much in there.
Okay. Now listen, I will explain. So in this lab, the white is your plasmid DNA, and the green or yellow is your GFP gene from a jellyfish. So this lab is super easy. You just follow the directions. You're going to be making one of these. You're making a recombinant plasmid, a mixture of GFP and then your plasmid DNA. That's what you're making. And you're going to put it all together. So it's going to, if you look on your sheets here, like the white one, notice there's one section that's bolded. And on the green one, there's 2 sections that are bolded, not the highlighted, but the bolded sections. The bolded sections are the restriction sites. That's where you're going to cut with the restriction enzyme. Your restriction enzyme is your scissors, okay? The like highlighted parts on the GFP gene are analogous to like the start and the stop for translation. It talks about it in the lab. You're not going to translate, but anyway. So you just follow the directions. It's going to tell you to cut. And when you cut, you literally cut along these like lines. So to see the plasmids, you get rid of all the extraneous stuff, okay?
I was thinking bigger than that. Like some of them are bigger. Bigger than that, small ones. Like it goes from bracelet to headband. Oh, can I borrow one of your viruses? Can I borrow one of your viruses? I do a daily virus on Snap. Like to educate people on viruses. Like I did daily rhinovirus yesterday.
The ones that are longer are the correct ones. Wait, what do you do?
Okay,
I wasn't listening.
so
What they did wrong.
I was asking if I could borrow one of your viruses because like I do like a daily virus on Snapchat and like to educate about viruses. So like I did daily rhinovirus and
Oh yeah. You can. All you're gonna do is make this and then answer the questions. Okay? It should not take you long, so I will give you until what, 50? You can finish by then. And then we'll it's louder than normal, yeah. So they were saying when we did the closed blue the other day, it wasn't loud enough on the 2nd and 3rd floors. So maybe they're just making everything louder.
Oh, it was so loud in there. I could hear it after I came out.
I mean, some of the teachers weren't even like, they were telling us to go into classrooms before it even started.
Yeah, they really should have just started saying like on Friday or Saturday, they got to get classes. I don't win.
We are gonna have an unannounced one sometime in the next week or so.
That's good. They should have it at lunch.
Oh my God, that's the
It's like,
actual.
I mean, now that you're going to a lot of front doors, and so like, our kids can't get inside.
Oh,
And
no.
after last year, we were walking, I put in my entire class, they locked the doors and we were like, can we come in? They were like, no, not gonna happen. They said, you need to wait outside and close the doors. And we were like,
You could get shot. Right, what a Oh, sorry. Wait, okay, 50. Y'all involved? 50.
Okay, from the white paper. Oh, these are too small for my hand. Alyssa, we need to switch. I got little scissors. Somebody gave me.
What in the world is this person doing? Oh,
I gotta go back to there. You gave Alyssa
all right, before you take them together, you should ask me. You did this in public class?
I know
Oh, not like that.
You have to ask that. No, the important thing is you know.
Oh my God. Cassandra.
Yes, they do.
No. Yeah, y'all, we're just using or hand free. You're not using star one. And that is just for other types. AAG, don't use it.
We were not using.
You're just using hand free. Redirection.
The wish.
Alright.
So, what about like this?
That's a nice cut.
So what?
Alright, this is just. Alright, from the green. Are y'all going out to dinner tonight?
Not tonight, but we're going to Motor Supply on Saturday night.
Oh, happy birthday.
Where's that? I mean, what's that? Like,
Motor Supply, it's on Downtown Gray Street. Super nice restaurant. It's different every day. I'll show you.
Oh, we never told you about Old Chicago.
Oh, how was it?
I didn't like it. I mean, I did not like it.
It's like, oh, there's enough stuff.